Endothelial cells line all the blood vessels throughout our body. But although it's a single cell type, the endothelial cells in my brain are different than the ones in my lung and different than the ones in my left toe. But how different are they? And how do we even study this? Well, let's dive into Chapter 2, the proteomic landscape of in vitro cultured endothelial cells. Before we head into the chapter, I have to introduce the scene that we are working with. So that our endothelial cells are different throughout our bodies has been known for quite a while. Endothelial cells are really capable of adapting to their microenvironment, and as such they are different in each organ. Large comparisons have been done using single-cell transcriptomics in murine models. So in humans it's quite a bit more difficult to get your hands onto fresh endothelial cell material. So we are more dependent on in vitro culturing of these cells. The problem with in vitro culturing is that, well, as I said, endothelial cells are highly capable of adapting to their microenvironment. So they will also start adapting to this plastic microenvironment and therefore slowly lose their organ specificity. But regardless, organ-specific endothelial cells are widely used in vitro as well. And you can buy a lot of different organ-derived endothelial cells for your in vitro cultures. So that's the current playing field. On the one hand, we have the marine transcriptomic studies, while on the other hand, we have the human in vitro studies. Which leaves us some questions. How does a mouse compare to a human in terms of endothelial cells? How does the transcriptome translate to the proteome? And how does in vivo translate to in vitro cultured endothelial cells? And to fully understand the nature of why our endothelial cells are different throughout our bodies, we need to be able to address these questions. So we wanted to try to add some parts to solving this puzzle. And we are a proteomics lab, so we looked at in vitro proteomes. Meaning that we acquired commercially all these organ-derived endothelial cells and also included some widely used in vitro models. Grow them and do proteomics. So the first point I want to make is that the proteomes of all these in vitro derived endothelial cells were highly similar. They are mostly overlapping. Now, there are some differences, and I want to start with this supplemental figure that I think is very important. So if we look at the PCA, which is a matter of distribution between all samples, or how similar or how different they are, as you can see, there's 3 samples separating from the rest of the samples. And it would be very nice if these were indeed of one specific organ, but actually they are a lung, a kidney, and a cardiac-derived endothelial cell. And it turns out that these 3 endothelial cells all came from one of these companies. So they are endothelial cells. I don't doubt that they come from the correct organ, but they are clearly combining together, which means that the isolation process depends the final proteome of this endothelial cell and potentially their phenotype as well. Now let's remove these and look at the other cells. Again, these are very similar, but we can sort of see 3 groups in this. One is the HUVECs, the other is sort of BOECs, livers and cardiacs, and the other group is the brain and the kidneys. However, you can already see, for example for the lung, that these replicates are very far apart, or even one of these livers that is completely different from the other 2. And also in terms of organ-specific genes, we see very limited proteins that are evidently specifically regulated per organ. If we look at the overall processes that separate all these cells, we mostly see a generic difference between smaller proliferating cells versus larger cells that enrich more for extracellular matrix processes. But in terms of organ-specific processes, it seems limited. There's one module here that is upregulated specifically in 2 out of the 3 liver donors, mostly extracellular matrix proteins. We also have one cluster that is specifically downregulated in the HUVECs, which enrich for tight junction molecules. And the final result I want to share is that we also checked the interferon gamma response in all these different endothelial cells. And interestingly, it is nearly identical, which highlights a very conserved interferon gamma inflammation response regardless of the endothelial cell type. So what do we learn from all of this now? I think it's too simple to say that my brain endothelial cells all are the same and have this specific characteristic. But instead, it's highly dependent on the microenvironment within that organ. So things like vessel size, whether it's oxygen-rich or oxygen-poor blood, the flow, the neighboring cells, the presence of different cytokines, all these building blocks will determine what phenotype the endothelial cell has. That our endothelial cells throughout our bodies are very different, there's no doubt about that. But how all these different building blocks contribute in what way to determine the final phenotype of each specific endothelial cell, that is something we are still very much at the beginning of figuring out. Which at the very least leaves something to explore. So as I'm filming this, I'm in the final stages of completing rebuttal 1. Let's see, I'll put it here, how long it will actually take before this paper is out and you will see this video. Who knows, might be a week from now or half a year. As for now, take care of yourself, take care of each other, and have a wonderful day.